Good morning, students. Today we discuss about the genetics unit 5th. Unit 5th is service related to genetics. In this first topic is genetic testing. Genetic testing is genetics analyzes your DNA, chromosomes or proteins to find changes, mutations or variants that can diagnose disease, predict risk, guide treatment or understand ancestry by examining samples like blood or saliva for inherited traits or cancer-related tumors mutations, offering crucial info for personal health and family planning. Okay. In this types of genetic testing, first is germline testing. Test inherited DNA passed from parents, and second is somatic testing. Test acquired DNA, often in tumors for cancer treatment. Next is clinical purpose. In this first, diagnostic testing confirms a genetic condition in symptoms individuals. Example, cystic fibrosis. Second is carrier testing. Identifies carriers of recessive genes mutation. Example, sickle cell, Tay Sachs. Then prenatal testing. In this, detect genetic conditions in a fetus. Example, Down syndrome. Next is pre-implantation genetic testing, PGT. Screens embryos for genetic issues during IVF. Then predictive pre-symptomatic testing. Check for mutations that increase future risk. Example, Huntington disease, some cancers. Next, pharmacogenomic testing. Guide medications choice doses based on genetic response. Newborn screening screens infants for treatable conditions right after birth. Then forensic testing used for identification in legal context. Then interplay. Genetics and epigenetics often work together with changes in one influencing the other. Then blood disorders. Screening can detect conditions like sickle cell diseases and beta thalassemia. Then next topic is gene therapy. Gene therapy in genetics is a revolutionary technique that treats or prevents diseases by modifying a person's genetic material, aiming to fix faulty genes, replace them with heavy copies, or introduce new genes to restore normal cell functions, often using modified viruses as vectors to deliver DNA or RNA into cells. Then methods. In this two types of method, in vivo and ex vivo. In vivo, delivering genetic material directly into the body. Example, by injection. Then ex vivo, removing cells, modifying them in the lab, then returning them to the body. The next is genetic counseling. Genetic counseling in genetics is a process where specialists help individuals and families understand genetic conditions, their inheritance risk, and management options. Integrated medical, psychological, and familial impacts to support informed decisions about health, family planning, and testing. It involves assessing family history, explaining genetic testing, interpreting results, and offering support, crucial for conditions like inherited cancers, birth defects, or prenatal concerns. Then next is benefits of genetic counseling. People with a family history of genetic conditions, example cystic fibrosis or Huntington's, and those planning a pregnancy or expecting a baby, especially after age 35 or with previous loss. Individuals with positive prenatal screening or ultrasound results. Then people with a personal or family history of certain cancers or birth defect. Okay, so today we discuss the genetic testing, types of genetic testing, then their clinical purposes, then gene therapy, methods of gene therapy, then genetic counseling and benefits of genetic counseling.